I'm going to represent the relationship between autonomic neurocardiac function and depressive disorder. This is my team. As you can see, we have just 3 members. It was hard to prepare this representation. Give our team some advantages.
Professor, please!
This is our content for today's announcement. First, introduction. In this part, I'm going to talk about why did we select this subject and explain physio-EX and HRV simply. Second, participants and method. Third, result. And fourth, discussion. And last, conclusion. We experimented about heart and autonomic nerve in the physio-EX that heart rate slow when the parasympathetic nerve is activated. We found that some of neurotransmitter, for example dopamine, influence mood. And also we had curiosity how they influence our body. So we thought sympathetic and parasympathetic nerve of many signals passing the neurotransmitter are associated with major depressive disorder. Therefore, we searched through paper the relationship between autonomic neurocardiac function and depressive disorder. This is PGOEX experiment that related with the relationship between heartbeat rate and vagus nerve. Look carefully the frequency of heart activity graph and red circle. You can see at normal state heart rate is 62, but when we do vagus nerve stimulation, heart rate is 20, slower than normal state. By this experiment, we can predict when vagus nerve stimulate heart rate is decline. Then, what is HRV? HRV is heart rate variability. HRV is predict changes in heart rate over time. It is related with autonomic nerve system that affect sinus nodes. So, HRV measurement method is to keep track of homeostasis mechanisms of automatic nerve system. While heart rhythm is regulated entirely by the sinoarterial node under normal conditions, heart rate is regulated by sympathetic and parasympathetic input to the sinoarterial node. The accelerant nerve provides sympathetic input to the heart by releasing norepinephrine onto the cells of the sinoarterial node, and the vagus nerve provides parasympathetic input to the heart by releasing acetylcholine onto sinoarterial node cells. Therefore, stimulation of the acylation nerve increases heart rate, while stimulation of the vagus nerve decreases. Then, what is HRV? HRV is heart rate variability, and HRV is periodic changes in heart rate over time. It is related with autonomic nerve system that affect sinus nodes. So, HRV measurement method is to keep track of homeostasis mechanisms of autonomic nerve system. Heart rate. While heart rhythm is regulated entirely by the sinoarterial node under normal conditions, heart rate is regulated by sympathetic and parasympathetic input to the sinoarterial node. The accelerant nerve provides sympathetic input to the heart by releasing norepinephrine onto the cells of the sinoarterial node, and the vagus nerve provides parasympathetic input to the heart by releasing acetylcholine onto sinoarterial node cells. Therefore, stimulation of the accelerant nerve increases heart rate, while stimulation of the vagus nerve decreases. HRV is measured by calculating the time between R spikes on an ECG trace. You have to notice that it doesn't mean changes by the time of heart rate, but reflects momentary heartbeat and the variation of RR interval. It means small variation between single cardiac cycle and next cardiac cycle. The following illustration is an example for HRV. You can see RR intervals a little bit different. The first part is 845 and second part is 745 and so on. This is HRV spectrogram. If interval of heart rate is measured in real time, it may be represented to real time per minute heart rate by the procedural gain. HRV spectrogram is graph changes of the heart rate calculated from measured heartbeat interval. The top and bottom spacing of healthy people is wide and regularly beats within a certain range. The top and bottom spacing of unhealthy people is narrow, as you can see. Now, we are going to see participants and methods. 30 patients who are aged 25 to 65 with major depressive disorder were compared with those of 30 healthy controls. We measure HRV parameter by SA2000E, analysis time and frequency part. Standardized HRV tests enable quantitative estimation of autonomic nervous system function. Let's see Table 1. Demographic data of patients with major depression and normal control subjects. SBP, systolic blood pressure, is 124.3 in patient group and 122.8 in control. DBP, diastolic blood pressure, is 76.7 in patient group and 75.2 in control group. You can see SBP and DBP are not that different between two groups. Let's see Table 2. Heart rate variability in patients with major depression and normal control subjects. After controlling for age and gender, subjects with major depression showed a higher heart rate and significantly lower modulation of cardiovagal activity compared to control. The total power PP band, very low frequency band, low frequency band, and high frequency band are significantly reduced in subjects with major depression compared to control subjects. HR means heart rate and SDNN, the standard deviation of the normal to normal interbeat interval. RMSSD evaluates parasympathetic nerve. PP reflects autonomic nervous system ability. VLF related with the thermoregulation. LF evaluates sympathetic nerve. HF related with respiratory activity and evaluates parasympathetic nerve. LF-HF ratio reflects balance of autonomic nervous system. In research results, patients with major depressive disorder represent higher heart rate than the number of normal control. That is to say, lower activity of the parasympathetic nerve seems to have relation. Decrease of SDNN means loss of ability to cope with stress factors and to control the body's autonomic nervous system. Decrease of the PP means decreased ability to control the autonomic nervous system, and it means also decrease in depressive patients. Contrary to expectations, LF decreased. It looks like because patients were so tired and lethargic that lost energy in body while testing HRV. Decrease of the HF means that HF isn't decreased well in normal persons, but in view of decrease in patients, we can find that depression has a strong influence on respiratory activity. But LF-HF ratio was equal. It looked like because the patient's mental and physical state affected while testing. In the case of LF and LF-HF ratio, difference resulted appeared, but it is considered because of passing over seriousness of the symptom having depressive patient. Impressions problem in the in the case of LF and LF-HF ratio, different results appear, but it is considered because of passing over seriousness of the symptom having depressive patient, imprecision problem in the examination, and problem for equipment accuracy. In view of these points, it should be further conducted various studies. Later, we need study whether disorder of cardiac autonomic nervous function is back to normal after getting better depression in depressive patients, and how antidepressants influences accommodative dysfunction of autonomic nervous function associated with depression. In conclusion, depressive patients showed increase of heart rate and decrease of HRV, and sympathetic and vagus nerves imbalance seems to be related in the high cardiovascular mortality partly. Depressive patients represent the disorder of autonomic nervous function in heart, as balance broken, ability to control sympathetic and parasympathetic nervous activity is reduced, and so it seems to higher cardiovascular mortality. This is the end of my representation. Uh, Thank you for listening.